Hello, and welcome to our next discussion in Small Ruminant 101 about genetics. Now, genetics are the blueprints that show and tell us how your animal is going to operate. The genetics behind your animal are important no matter what your goal is with selection. All body cells contain these blueprints with instructions as to how an animal will operate, how it will look or act. DNA contains the genes of traits in a double helix ribbon located in every cell. DNA testing is something that is available. Not many people know about this, but in animals, you can certainly do DNA testing. UC Davis, the university there, does DNA testing. Pedigree International, Biogenetic Services, Genomes, and VetGen all do DNA testing. And that is important because it can let you know if you don't have any data on your animal, don't know previously what has gone on with your animal or the genetics. You can do some DNA testing just to find out what you could expect, what genes that animal might be carrying. A combination of genes are attached to chromosomes which guide the animal to express a trait or response. So DNA is the home for a gene. All species tend to differ in the number of chromosomes that they have. As you might know, humans have 23 pairs of chromosomes, cattle have 30, sheep have 27, swine have 19, chicken 39, goats have 30, and horses have 16 pairs of chromosomes. And we will see why that's important here in just a few minutes. Now, I'm going to take a few minutes here to define a few terms that will be important as we go forward. We'll talk about the differences between heterozygous and homozygous. But before we do that, we need to talk about what alleles are. They are a variant form of a gene. Humans, as well as small ruminants, are considered diploid organisms because they have two alleles at each genetic locus. And so what a genetic locus is what the genetics are going to look like or what the genetics are going to do. And so humans and small ruminants have a pair of alleles at that locus. So a heterozygous animal means that the genetics or the genes at that locus are the same or are different, excuse me. And for a homozygous, it means that they are the same. Another important thing that kind of goes along with the homozygous, heterozygous is gene dominance. This is when one gene overshadows the expression of its other allele. So as you might see in this picture here, white is a dominant color in goats. WW and W small w are both going to be expressed as white because that W or that white is a dominant gene. Recessive genes, on the flip side of that, they're overshadowed by that dominant gene. They can only be expressed when they both are present. So for example, in goats, red coat color is recessive. So in order to have a red animal, you would have to have small w, both recessive genes are present there, so it would express as a red animal at that point. So genotype is just the genetic makeup of an individual animal. So for example, big W would be a white animal, big W would be a white animal, little w would be a red animal. And so we can use a Punnett square to help us predict the genotype of offspring from a pairing of two animals that we might be making. So in my little example here, I have two heterozygous white animals, white goats, for example. So I have big W, little w on top, and big W, little w on the side. Now, as you can see there, you just fill in the Punnett square by simply pulling those two W's together in each square. So our top left square, we have big W, big W. Our top right square, we have big W, little w. Our top left, or our bottom left square, we have big W, small w. And then in our bottom right hand, we have small w, small w. So we are essentially going to have a 25% chance of getting a homozygous white animal, a 50% chance of getting a heterozygous white animal, and then a 25% chance of getting a homozygous red animal. In other words, we have a 75% chance of having a white animal and a 25% chance of having a red animal. As you can see, or as you should remember, white is the dominant color, and so it is harder to overcome that. So the odds are stacked against us there. 75% chance of having a white animal, only 25% of a red. Phenotype: an animal's visual expression or performance in any trait. Phenotype is the outcome of genetic transfer as developed in a production environment. Phenotype defined as the sum of the genetic and environmental variation. The genetic variant accounts for only 25% of the phenotype, while the environment accounts for 75%. If you couldn't tell from that, environment is going to play a big factor in how these genetics are going to express themselves. So some sources of environmental variation are as follows. Weather. Just think about maybe earlier this spring coming out of winter, we were having some days where it would be really cold and rainy, and then a few days later it would turn up and be pretty warm and dry off for a few days, and then we'd go back to snowing. The weather can play a big factor in how your animals are going to thrive in their environment, and that's the number one thing to consider when we're thinking about environmental variation. And when you're thinking about selecting animals or raising animals, you need to really look at the weather in your area or your region and determine what you would need to select for. That way, environmental variation as concerning to weather will not be as big of a factor. Another factor is land topography. This is just like the lay of the land. Thinking about if your animals are living on a hill, flat ground, in a river bottom type area, just keeping that in mind, you'll have to make sure that you factor that in when you're thinking about how these genetics that you're going to be selecting for are going to express themselves. Structural resources. These are things such as barns, places to get out of the weather, places that forage will be stored or available to animals if they're having to drink out of a creek, or if they have tanks of water that they get to drink out of. Those structural type of resources, those management type of things. Another one is their forages and feeding. You need to think about if your animals are going to be out on a nice fescue pasture. Are you going to have these animals in a dry lot and feed them? What kind of forage are you going to be supplementing with? These are all things that can vary your genetics and how they will achieve their highest or their fullest potential. And then ultimately, health and sanitation is going to be important. If we're keeping all of our areas clean of fecal matter, if we're making sure animals are up to date on shots and dewormers, those all will play a factor in how those genetics will express themselves. So I have another scenario for us to consider here. Which buck has the best genetics for growth? Now, this is a little bit different if you tuned into our Small Ruminant 101 on selection. If you remember earlier, we were selecting for who had grown the best, or which animal had grown the best. Now we are selecting for which animal has the best genetics for growth. And those are two different things. From this picture, you might say the buck on the left-hand side is bigger. I would probably agree with that. But we have to take into account the environmental factors. Does that necessarily mean that because the goat on the left-hand side is bigger, that he has the best genetics for growth? No, it does not. The buck on the left-hand side could have been kept inside in a heated and cooled barn with full feed and the best forage and cleanest water around. And the buck on the right-hand side has been turned out to pasture ever since he was weaned and has only eaten fescue grass. Maybe the buck on the right-hand side was the one that was put up in the barn and fed the best, and the one on the left is the one that went out to the pasture. We don't know those environmental factors, so it's hard to make the decision on which one has the best genetics for growth. The only way we could do that is if we made sure both of these animals were put into the same exact environment, and then we could determine which had the best genetics for growth. Improvement of any trait is going to depend on measuring differences in a trait, accurately identifying the superior individuals in that specific trait, and then using those superior individuals as parents, and how heritable those traits are. This is important when talking about genetics because we need to make sure that we are picking the animals that are doing the best, are superior. We're also thinking about how those genetics and that the different generations have performed, and using that when we are selecting animals to use as parents for our next generation. A sound breeding program is going to make sure that we have maximum genetic improvement. And we have to make sure that we are allowing those improved genetics to be expressed. So if we are breeding animals to grow faster, and we are selecting those sires that are going to give us the best growth rate, but then we're not putting those animals, those kids or those lambs, in an environment for them to grow, if we're not cleaning out their pens daily, or they don't have enough forage or feed to actually grow, then we're not going to make any improvement. We want to make sure that we are giving an environment to those individuals for that information to be expressed. Now, I want to talk a little bit here about something slightly different, but certainly involves us thinking about genetics, and that is some livestock breeding systems. So when we think about that, there are four that come to mind. We have grading up, crossbreeding, inbreeding, and line breeding. And we'll briefly go through each one of these and figure out the differences and maybe look at some ways that you could utilize those as a producer. Grading up. This is breeding purebred sires to commercial females. Replacement females are kept and bred back to the purebred sire. Your aim is to produce progeny that resemble the purebred sire. This happens in the fourth generation. That's when the most improvement can be realized. So grading up is taking your commercial females, finding a purebred sire that has some genetic information, some pedigree that backs up how he is going to perform, how those kids should perform, and then using those replacement females and bringing back to that same sire. We're going to get that fourth generation and notice those traits really starting to come out that we are selecting for. Crossbreeding. This is a very common breeding system that's used. This is the mating of animals of different breeds. Your aim here is to take advantage of both hybrid vigor and breeding value. And so hybrid vigor, also termed as heterosis, is crossbred offspring exceeding the average of the two parent breeds. So we are putting two different breeds together, and hopefully what we're putting together, that offspring is going to exceed what those two parental breeds would have reached by themselves. Hopefully that makes sense. Crossbreeding is one that's used a lot in all different species of livestock, and certainly we can see a lot of improvement quickly in these animals. Inbreeding. It's a mating of related individuals, sires and dams which share at least one ancestor. This is most commonly found in the purebred industry when they are selecting those genetics and those matings to make them look on paper really good. We want to see an ancestor that is maybe highly sought after on both sides of that pedigree. Your result, you're going to increase some uniformity of the offspring, but you might also have some unwanted genes surface. So because those genetics are getting closer, there are unwanted genes that are typically recessive, but when you mate animals that have those same recessive genes, as we saw in our Punnett square earlier, your percentage chance tends to rise. So keeping that in mind, you want to make sure when you're inbreeding that you don't inbreed too much, that you do make an outcross at some point to bring some new genetic information in. Line breeding, it's also another form of inbreeding, but this is with an attempt to maintain a close relationship with a highly regarded ancestor. And so this is most found in the purebred industry again when we're thinking about that paperwork. We're going to breed maybe an elite sire to some half sisters. We want to keep those genetics as close as possible to that elite sire. And so we have those same parents, and we're just really wanting to express those traits that that elite individual has in those kids or those lambs in that offspring set. We want to see that elite individual hopefully passing on a lot of those same traits because of how close in relationship the sires and the dams would be. So in closing, and we've talked about this throughout both of our discussions, but the single most important trait in the goat industry and the sheep industry and any livestock industry is certainly going to be reproduction. From a selection standpoint, we talked about how if there's no product for our producers in reproducing kids and lambs, there's no product to move down the line to the other people. In our genetic discussion, we talked about reproduction is important when we're selecting genetics that we want to see expressed, because if without that reproduction, then we aren't going to see those individuals and those genetic pairings expressed. So reproduction is very important and something producers should put a lot of emphasis in. That's all I have. If there are any questions, you guys could certainly email me, LHICKER, then the number 2 @utk.edu. Thank you.